Yeah, I will show you. I am running, that may be important for some of you. With session info, I can know which version of AIR I'm running, and I'm still on AIR 3. So since a year, it moved to AIR 4. I haven't moved yet to AIR 4. So of course, as David says before, in 2 hours it would be a bit too short to go in deep in all the commands. So some parts will probably be too quick to you, but just ask question in the chat. Of course, the code I show, it's my own flavor. There's no gold standard way to code. There's multiple other ways to code efficiently in R. So you will have to adapt to your own flavor if you are not happy with my coding. And last thing is that I won't use packages which are actually designed especially to analyze meta-barcoding dataset like Philosec, which makes things really easy, but you don't really understand what's happened in the background. So what I want to show you is the low-level summary statistics of meta-barcoding datasets. So we can start with loading packages. So like I will use tidyverse a lot, which include ggplot, db-RDA, table, and so on, many other packages. So when you load a package, it's really important you keep in mind that there's an order in which you have to load the package, because if you, for example, need the clear package, you will see some warnings says it's overwrite all the function or masking all the function. So, and in my case, summarize or arrange. So this function, I prefer to have it from tidyverse than from clear. So here is the wrong order to load the library. I need to load library clear first, and then tidyverse second to use all these functions from the tidyverse packages. So if you are in that case and you load the libraries in the wrong order, you do session restart air. It will back up all your environment, just reload it if you have any, and then you can start to reload the libraries. So now we load clear first and then tidyverse, and now tidyverse say it will overwrite some of the clear function like mutate, summarize, and so on. Another cool features in RStudio is to create paragraphs in your script. So with Control-Shift-R, you can create sections. So like we will start loading the data, so we create a section load data. It annotates the scripts with some fancy minus, and here at the bottom of the scripts, you can quickly access these different sections. So you will see later when you have 10 sections, it's make it easy to travel through all your scripts. We'll start by loading the OTU or the ASV table, which is in community matrix. So it's CSV file, but in fact, it's a semicolon separated value. So we use read CSV2, which use semicolon as separator and not comma or column. And we use the first column in this matrix as row names. So you can load that matrix, you see the element is loaded in your environment. And what's important is to know what's inside your object. So you will have to use multiple small functions to look at what is in your object. So mat, here it's 65 rows and 399 columns. You can also use structure, the function str, which tells you the structure of your object. So it's integers with dim names. So the row names are the names of the sample, the column names are the names of the ASVs. So now we can load the taxonomy, which I store in the taxo object. And because I'm in R3, by default, when you read the data frame, all character columns will be translated to factors. If you don't want that in R3, you have to use this option. In R4, it's by default false anytime. No. So you don't need that. Taxo, one good way to look at your object is also to do head. So here you see that head doesn't help much because you print also all the sequences. One column is sequence, so it's not so nicely displayed. And you see also that the taxonomy is not splitted, so it's not easy to manipulate. But we will check that later. At least we have load the data correctly. And we can load the environmental data with the same command. And again, check the content. So in env, the row names, the sample names, and then we have some environmental variables. So now we want to control the data and perhaps edit them if we are not happy with their format. So we create a new section, control data. And then we need to control whether we have the same ASVs and samples in all the three tables. So the dimension will tell us the matrix have 65 samples, 3,991 ASVs, 65 sample in the environmental table, 3,991 ASVs in the taxonomy table. So on the dimension, we are fine. We can also check that the names of the samples, they are equal between the matrix and the environmental table, which is fine, and between the call names of the matrix and the ASV IDs in the taxonomy table, so the first column. And here you see that all of them are mismatches. Why that? It's because here the ASV ID are named with ASV 1, 2, 3, 4, 5, and so on. And if you look at the call names of the matrix, you see that in the ASV matrix, you have the number of the ASV plus underscore symbol plus the number of reads of this ASV. And that's why you have a mismatch between all of the names. And for that, you need to correct the column names of the matrix. So you can also look at that with heads, so as written in the script. So again, we see that the column names of matrix are different than the column ASV ID in the taxonomy table. So we need to take care of that. So actually, we can use the substitution function to remove all these parts in the column names of the matrix. We can go back up, see the column name of the matrix, how they look like now. And we are fine. So we can test whether it's all equal, and now we have exactly the same naming for ASVs in the matrix and in the taxonomy table. So we have controlled the data are good. We can save the script and go to the next step, which will be summarizing the data. So first we can have a look at the read count distribution. So we can sum the number of reads in each sample, so the row sum, and summarize it. So we see that we have from 24,000 to 780,000 reads per sample, with an average at 400,000. We can also see that one sample, the GR2, have almost 25,000 reads, and the next one with the lowest number of reads have more than 100,000 reads. So this one is relatively low amount of reads. And we can actually consider removing this sample from the analysis. And for that, we can actually look whether we have enough replicates for that. So in the env table, actually, I'm going a bit further in the script where we summarize the environmental parameters. So actually, I'm now counting how many samples we have per habitat and per peatland. So as David says before, we have the data from Protist in peatlands in 5 different habitats and 5 different locations, so peatlands. And you see that the one peatland have 5 replicates of each habitat, and the other have 2 replicates of each habitat. GR2 is the row name. You can subset the nth table by row name. It's from this Gruyère peatland. So we have only 2 replicates of the lone habitat. Actually, it's not ideal. Removing it means that we will have only 1 replicate remaining. But in that case, it means anyhow we have only 2 replicates, and we cannot do the analysis inside each peatland. So we will have to do overall analysis over the different habitats to have a statistical power. So 1 replicate less or more won't impact much on analysis. But we will see later. So this sample we can consider to remove it. So another quick summary statistic we can do is RADFIT, which is computing the rank abundance curve. So of course you don't find the function because it's in the vegan package. So we have to go back up. So I typically always write in the first line all the package I'm loading. So every time I reopen this script, I just load all the package needed to execute all function of the script, and I'm fine. And here, there's no warnings that function are overwritten or masked, so no interference between the different packages. So RADFIT, it's actually doing a rank abundance curve. So the y-axis is a number of reads in a log scale, and then actually RADFIT try to fit some functions to the curve, which is not really interesting for us. But here we can see that there's almost no singletons, so ASV, which have only 1 read, and that's actually a consequence of the ASV calling. Data2 is typically removing all singleton ASVs. So that's what you can read also in the script. If you do again the call sum, that means the sum of all reads from each ASVs, and you sort them increasing order, and you do some table statistics. So you see there's only 63 ASV with a count of 1. And then as soon as you go to count of 2, then you again increase the number, and then it will again go decreasing in terms of abundance. You can do that also at the sample level. So here in the first sample, you see, of course, there's many ASV not occurring in that sample. There's only 8 ASV with count of 1. Few have 2, few have 3, and so on. And then you can plot the rank abundance curve also at the sample level. So yes, for 1 sample, you see that the form of the slope is relatively similar to the form for all samples together. So we do have singletons at the sample level, but not at the study level. So it's due to ASV calling. Due to that, if you do rarefaction analysis, you will most likely have saturation of diversity because you don't have much low abundant ASVs, and it's probably better like that to remove low abundant reads because they could originate from tag jumps, but we will take care of them a bit later. Now we can have also a look at the taxonomy. So again, we have in the taxonomy the ASVs, number of reads, person identity, the best match in gene bank, so the gene bank accession number, the taxonomy, which is separated by pipe symbols, and the sequence, so the reference sequence. So it's not really easy to manipulate the taxonomy as a one full string, so Eukaryota and then Streptophyta, Embryophycea, and so on. So we need to split that taxonomy. So I use a loop here, which loop on the ASVs, actually on the column, and it will split the gene bank taxo field in multiple So here I control the length of this taxo list. So how much rank in the taxonomy we have in this GenBank taxo. And actually it's not GenBank taxo, it's PR2 taxo, because GenBank would have been with a variable number of taxonomical rank. So in GenBank, not all taxa have the same number of taxonomical ranks. Some protists have only 6 ranks, so they have a phylum and then class, family, genus, species, and that's it. And some plants will have until 25 taxonomical ranks. Makes it difficult to handle in R. But here with PR2 taxonomy, we have 8 fixed rank, and we can use them easily to recreate our taxonomy table. So we will create this ranks vector, and we will assign this vector of ranks to the different taxonomy. So now I will recreate the taxo object by reading again the CSV file, and then I use the piping operator. So that's the piping operator used in the tidyverse package. That means the output of this function is sent as input of the next function. In R4, you have now a native pipe, which looks like, I think, superior straight bar. So you don't need to use the one implemented in this package anymore if you use R4. So now I want to redo what I've done before, but in a more compressed, tidy way, and then rename all the columns. And now we can have quick view on the taxonomy. So you see now we have 1 column per taxonomical rank, where data frame is made of factors for the taxonomical ranks. And you can summarize already the phylum level. So you see that we have mainly fungi and metazoan, and then cercozoan ASVs. It's the number of ASVs. That doesn't mean it's the amount of sequences. So again, back to the experimental design. So as I told you, experimental design, we have different peatlands and 5 habitats in each peatland. 2 to 5 replicates, and then 4 environmental parameters we measure. Actually, it's easier to manipulate such data frame when the samples are not in row names, but as a column. So that's why I reread the input file and then have the sample name actually in a column. So you will see later we will use that. So now we can summarize again with count by peatland and then habitat, or by habitat in peatland. But we have seen that already. I can show you quickly how we correct the data. So it's levels, peatland. And yeah, as I've read in the script, cow, it should be actually Cashew, it's the official name of this peatland, so we can modify that when we read the table. There is some function from the forcats package, which is loaded with tidyverse, which allow you to manipulate factors. So if you want to recode, relabel, you can change the name of a level. And it's better always to do it at the beginning, so to name all you need correctly at the beginning, so that you don't need to redo it every time you plot a figure or you do some table summary of your data. So now I rename the peatland correctly, and I change the order of the different habitats so that they are in logical order. And now we have cashew, and then the habitats are from forest, actually transition, I think, before forests, lawn, hummock, pool, which can be a transect through the peatland. So now I would just copy paste this paragraph on the hypothesis to test. And again, you see the section. Now you see the number of sections expand to 4, so it's easy to navigate from one to the other when you have this section delimitation. So what do we want to test quickly? So on the alpha diversity, we want to know which habitat is the most diverse, but the bias we will see is that there is a sequencing depth bias. We also want to know which taxa is more abundant in which habitat. The problem is that when you compare sample, then you compare compositions. So the values you get in your matrix are proportions of each ASVs in one sample. And if one sample have 10,000 reads, so 1,000 reads doesn't represent, it will be 10% of the sample relative abundance, while 1,000 reads in a sample with 100,000 reads, it's only 1% of the total relative abundance. So that is composition. So we are dealing with relative abundance and not real abundances in our samples. And we need to take that into account when we do a comparison of relative abundances. And then on the beta diversity level, that all samples are similar to each other or dissimilar. We want to know which environmental parameters explain most difference in community composition or structure, but mainly at the Protist level. And here we have all the bias, like environmental parameters are not normally distributed, and again, the compositionality, which can be a problem. And of course, we have mini pitland. So if we build a statistical model, we will have to add pitland as a random effect. Just like if you have multiple transects from one habitat, then you will have to add transect as a random effect in your statistical analysis. Well, so now we need to filter the data. So because we have used this general primer, and we not only have protists in the data, but you see metazoan, fungi, and plants. So let's go to filter the data. So one first step would be to remove sample with lower read counts. So the sample with low read count we have seen before, and also because we don't have any way to check for tag jump issues. Typically, tag jump will happen for ASV with relatively high number of reads, so they have more chance to produce tag jump and end up in the wrong sample. And every ASVs with low abundance in one sample are actually questionable. So you don't know if the origin is biologic or due to tag jump or to artifacts. So if you have no idea about tag jump in your samples, one safe way is to remove ASV which occurred in each sample less than a certain threshold. Let's say here we use 4 reads, it could be 10 reads, it could be 100 reads. There's no real ground truth of how to set this threshold. The best would be to have positive controls so that you can control for the tag jump, as we discussed already yesterday. So again, the idea would be to remove this GR2 sample so that we have only one lone from Gruyère remaining, lone habitat. So we first begin by removing this sample from the environmental table. Don't forget to use drop level if you have factors in your table. And then we will create this abundance matrix with the occurrence of 1 to 3 reads. So I create a temporary object mat_abi, and then for all values which are 3 or less, I just erase them and put 0 in place. And from this matrix, I am also removing the sample I have removed from the environmental table. And then I pipe that into a command which will remove all ASV which sum to 0. Some ASV were perhaps only found in GR2 samples, so we need to remove them from the matrix. They are not useful anymore. And then we can compare what was the effect of this filtration. So we remove one sample here. We remove one ASV, probably ASV, the only ASV which was only found in GR2 sample. And then we lose some 25,000 reads due to the removal of low abundance ASV occurrences. last, we need to filter the taxonomic table to remove the ASVs, and we are done. Next step is to filter the protists. So we don't have only protists, as I said before, but we have also plant, animal, and fungi in the datasets. And you will see that we have a lot of them. So here in this function, so mutate will create a new column in our taxonomy table. The column will be named taxa. And then to create this column, I use a if-else function, and actually a nesting of multiple if-else. So if the column phylum is metazoa or fungi, then label the taxa as metazoa or fungi. Else, then I use a second if-else. If the class is Embryophycea, which are the higher plants, just check if it's sphagnum. If it's sphagnum, then label it as sphagnum. Else, label it as embryophytes. And if it doesn't match any of this option, then label it as protists. So all remainings will be protists. Well, and then if you look at the object, so for all ASVs, we will have these labels, and you can already do the quick summary statistics. So you have actually most OTUs are protists, which is good. You have 31 ASV, which are sphagnums, and you will see that actually they represent most of the reads. So now that you have labeled all your ASVs with these main clades, can create a data frame from the matrix to know how many reads you have in each of these clades. So here I just transform the matrix into a data frame. I had the sample as first column, and then I transform the matrix into a long data frame. So for that, I really recommend you to have a look at the tidyverse cheat sheets and so on. So if you search to this link, if you look at that link and you search for data wrangling with dplyr and tidyr, then you find lots of explanation on how these functions are working. But just as a quick overview, here we have the OTU matrix as a data frame, and then when we pivot it, then we now have only 3 column remainings. And I will show you. And each line actually is the combination of the sample by the ASV and the read count. So we just transform the matrix into a really long data frame, 1 line per observation. Of course, you have many 0, so you need to filter them, and then you can add to each ASVs their taxonomy. So with left join here, as we have the same column names, it will use these columns to join the 2 tables together. And then we can also add the information of habitat and peatland by joining on the sample names. And then you can see now for each ASVs, we have sample ASVs, the read count, the taxa, the main taxa, the habitat, and peatland. So now we want to have overall read proportion in the different habitat and peatland. So what I'm doing here is that I'm grouping this data frame by habitat and peatland, and then I'm summing the reads for each of the taxa. So you have a sum of reads for each taxa in each habitat and peatland. And then you can type this input. So either you save that in an object or you pipe it into a ggplot. So I prefer to use ggplot because it's more easy functions to create plots. So here we want to have a peatlands, which will be the x-axis against reads, which will be the y-axis. So how many reads we have of each taxa in each peatland. And then we will create a bar plot with colors being, so the filling color being the type of taxa. We use stat identity. We don't want Guillaume to create any statistics on our data. and position fill will actually fill the bar on 0 to 1. So that all the bars will have the same size and we will show proportions. So I can already execute till there. We also wrap, so do facet wrap. So we have 1 plot per habitat. So we will create 5 plots. So you can already see that there's lots of purple, which is sphagnum. So as say David, he samples sphagnum samples, and there's a lot of sphagnum. So here would have been a great idea to use blocking oligos. Of course, this was done 10 years ago, so we had no ideas of blocking oligo at that time. But now if you sample on plant-associated or animal-associated communities, then it will be a good idea. And then you can improve a bit this graphic. So here the legend could go in the free space so that you expand a bit the view. You can label as %. You can orientate the labels of the x-axis with angle so that it's easier to read them. So that's all what I've done here in the next lines. And now the display is a bit better. So, and then if we want to only work with Protist, then we will have extract this blue part in all of the samples and only work with this blue part, which is like 1 to 5% of the reads in each sample. It's really low and it's not ideal at all. But we still have a good signal of the Protist community with that. So at least from the most abundant Protists, which will end up in this sequencing. So let's go and select the Protists. So it's fairly easy. Now which we have taxo eukaryota table, we just select the ASVs with label Protists, and then we select all these ASVs from the taxonomy table, which is taxo AB so far. We can do head again. So you have the same table as before, just that you only keep the ASVs which are assigned to Protist groups, Mycids, ciliates, Upalosa, Phyta, and so on and so forth. So once you have done that for the taxonomy, you have to do that also for the matrix. So you subset by using the SV names in this taxonomy table. Now we can summarize number of reads we have per sample. So we have from 5,000 to 43,000 reads per sample. So Protist reads per sample, which is far less, so from the 100 to 700,000 reads we had before, due to this co-amplification of sphagnum DNA. And actually on Protist, it's the same. We don't remove any sample, but I just create this object to remember I work with Protist data. And we can also have a look quickly on the rank abundance. We have a small error because one of the models did not fit correctly. Nothing seems to be too weird. We don't have a long tail of low abundant ASVs, most likely because we have co-amplified lots of sphagnum and we haven't covered most of the Protist diversity. If you plot rarefaction, which I will not show today, and that you can look at next package, I think it's like that, to plot rarefaction curves, you will see that the Protist diversity did not saturate. So let's move to alpha diversity. Finally, create a new section, alpha diversity. So yeah, I put that directly, all the functions. So by alpha diversity, I mean the different alpha diversity indices. So I will look at richness, use spec number function from vegan to get these values. Then you can get Shannon diversity with the diversity function, and then Simpson diversity with, again, same function, but saying the method is Simpson. You can also add the read counts of each sample and pivot that to a long data frame so that we can use it for plotting in ggplot later on. So we have this alpha table and we can do some quick plotting. We will do some quick plotting, but first I have to introduce you on the first big bias we will have to deal with, is the sequencing depth bias. So typically when we don't sequence enough, or when we have lots of co-amplification, like here, more we sequence, more we will cover diversity of our target group. So this bias is so important that most of the variance in richness is explained by the sequencing depth. So here I use this table alpha. I loop on the index, so that means I will slice this table using the index variable. So I will extract first all the rows with richness as index, and then Shannon, and then Simpson. And this slice will be temporarily stored into X. So the object is X. And with this object, I will create a linear model between the number of reads and the value of the alpha diversity index. So between the richness and the reads, between Shannon value and the reads, between the Simpson value and the reads. And as sometimes you get a better fit with a log transformation or square root transformation of total read count of a sample, I also have all these options. So I've got 9 linear models, 3 indices times 3 type of linear models. And then we can do some summary statistics in these linear models. So this one is a big long list of linear models. So it's hard to read just like that. So it's actually even a multi-dimensional list. So with 3 levels first by index, and then in each index you will also have 3 linear models. So using reads, log reads, and square root transformation of the reads. And here, so these comments will summarize actually the linear model. So the type of linear model and the index we use. And you see here the R square, so the percent explanation. And you see here that the log reads explain until 65% of the variance in ASV richness. The p-value is extremely significant. So we have a really big bias. And we can see that in a plot. So on the x-axis, we have the number of reads. It's, I think the x-axis is log transform. And on the y, we have the alpha diversity. And you see that more you sequence, or more you have sequence of Protist, more you have diversity. So that's, if you compare this sample at the top with this sample here, or let's say here the 4 samples are from one habitat and these 4 samples from another habitat, just if you're unlucky, then you will say, well, this habitat is much more diverse than this one in terms of Protist. But it's really totally wrong, because it's only due to the sequencing depth. And you see that on the contrary, Shannon and Simpson diversity seems to be linearly negatively correlated to the number of reads, which again here might be due to high amounts of co-amplification, and also, yeah, more you sequence, perhaps more you have chance to get sequence from the highest abundant Protist. Well, but all this variation is mainly technical bias. One way to handle that, so you can do rarefaction. That's one way to go. The issue is what with rarefaction is that you will take one random subset of your data set, and sometimes you lose like 90% of your data set because you need to take the same number of reads in each of your sample. And if you remember on our Protist math, so the sample with the lowest number of reads is 5,000. So here you will take only perhaps 8% of the reads of this sample when you rarefy it. So if you do rarefaction, my advice is to bootstrap the rarefaction multiple times and then run your statistical tests multiple times and do p-value corrections to be sure that you don't have only a special case of the random rarefaction that that you will observe significant differences, but only if you do this specific random rarefaction, and it will not work with other rarefactions. Another way I found a bit more elegant is to actually use the residual variance of this linear model. So all variance, which is not explained by the linear model. So all variance, that means the distance of the point perpendicular to this linear model here or there. It's supposed to be biological variance. So the residuals of this linear model then will be your biological variance. And you can actually scale them back on the -1, 1 scale. So in this column res, we have the residuals. So depending on the index, it could be relatively large. So richness can range from -100 to +100, something like that. And then you can rescale it on -1, 1 scale, so that we have the same scale for all the 3 indices. And just to illustrate, know that the residuals, so I plot again the residuals of the linear model against the total read count of each sample, or Protist read count of each sample, and you see that we don't have any more correlation, of course. And we are good to go, and we don't have any more this sequencing depth bias. So now we can summarize the alpha diversity per habitat. So I just add the environmental parameters, group the diversity by habitat and index, and then compute the average values. So we have one average alpha diversity value per habitat and index. And then we can test the change, so whether we have different alpha diversity between the different habitats. I have no look on the Zoom interface, so if people
just maybe one quick question: why do you have some red value in the mean here? It's not that common. Maybe you can explain.
That's the display of table. It's when you have negative values, and of course the alpha diversity is ranged. So the residuals, they are on -1, 1 scale. So some will be negative, some will be positive.
So it's true that with this transformation of diversity, your diversity can be negative. So it's not that straightforward at first, but it's a nice way to remove this artifact, as Guillaume explained. Yeah, but it's a bit complicated to understand the way to do that, but it's a very inefficient tool to remove this sequencing artifact.
Of course, if you're interested to quantify the diversity and to know how many species or OTUs you have in one sample, and say, okay, I have that much, to give a number, we are not using the right tool to estimate that due to this bias. Or you will have to sequence with V4. Actually, on terrestrial data sets, you will have to sequence 1 to 500,000 reads per sample to really saturate the diversity and give a proper number. Of course, you can try to do rarefaction extrapolation to see where the plateau is, where you have saturation. But still, this approach are biased by the low abundance ASVs or OTUs, which are typically removed with the bioinformatic approach. So it's really hard to say we have 500 ciliates in our pit bog. You never know exactly if this number is right, or if it's 10 times too much, or if it's 10 times too low. So you have an idea, but you can't really say a number. So what's matter is to be able to compare samples. And that the values range from 100 to 500 ASVs, or range from -1 to 1, as long as you can compare your sample average through samples and compare between type of habitats. Then, so here again, I won't go much in the detail of this analysis, so don't explain all one by one. But if you like, the best way to go is to split the data in smaller pieces and try to apply the function to only part of the data, so only to richness, or only to Shannon-Simpson values. What I do here is that I just add the habitat type to the alpha diversity values. So we still have our residuals, scale from -1 to 1, and then all the habitat informations. And then for each index, so here it's a loop, ddply, it's a loop which take a data frame as input and give a data frame as output. And it will again slice the table, the data frame per index, put that in this temporary object X. And then we create an analysis of variance. So we want to explain the variation in res, so the residuals of alpha diversity, by the type of habitat. And then we do a post hoc test, so a non-parametric post hoc test, the Tucker test, to test for significances. And then here's just extracting the values from the post hoc test, reformatting it to a data frame, relabeling it correctly, changing the column names, and so on. So that it's easy to manipulate. So now at the end we get that kind of output. So for each index, we compare every pairs of habitat, and we have difference, the amount of difference, lower upper difference, and the p-value. And then you can filter the significant difference. So in that case, we have only one significant difference between forest and pool habitat. I would say the forest is higher in richness, but we will see now in the figure. What is convenient when you plot a figure is to add letters, so to discriminate this habitat. For example, forest will be letter A and pool will be letter B, and all other habitat will be letter AB. So for that, I use multi-count letters. So here, definitely I won't go into the details. But for that, you need to load the package multi-count view. So go back up. And then in the case there's no difference, significant difference, then I put NA, so nothing will be plotted. And you see that forest will have A and pool a B, and the other AB for richness. Great. And then let's go for the plot. So for alpha diversity, the easiest is to do a box plot. So again, I add the habitat type information to the alpha diversity with a left join function. If you don't know how work left join, just go in the help. You will see that there's multiple kind of joining function. I'll let you, the best way to understand how it works is to go to the examples and try all the examples to really understand what it implies. So now I want to plot for each habitat the alpha diversity in terms of box plot. And I want to add the letters using alpha habitat letters on top of that. And of course, I do one figure per index. That's why I use the wrapping function to create 3 different facets. So let's go. So we have our 3 graphics, nice box plots per habitats. And then you see that here the pool have lower richness than the forest in terms of Protist diversity, which probably makes sense in terms of biology, because forest litters are much more diverse than aquatic environments. And for the alpha diversity, so the other indices, we don't see any significant differences. And then if you really want to be fancy and have fancy graphic into the PDF, just save them with ggSave. You can adjust the width and heights multiple times so that you get the size of all the text correctly. So the higher the figure is in terms of yes, inches, I guess, the lower the text will be written, and the other way around. Great. 1 hour. And we are done with alpha diversity, so now let's go for...
just a quick question. What's this 10 row containing missing value, the warning message you got at the end?
That's the NAs. So it's when I plot the letters. And I take profit of this function of ggplot. When you see NAs or missing value, you won't plot anything.
Yeah,
so
thank you.
It's a way to plot letters here and not plotting anything here. If I had put zeros or A everywhere, then it will have put A everywhere. Actually, you can also filter. I could have filtered the table. So let's try that. You can filter you can remove all the rows with NAs in the column letters, and then you won't complain anymore. So it's the same plot, but yeah, you remove NAs. But it's only a warning, so you don't really care. But it's cleaner like that, you're right. So let's go to the next section, taxonomic composition. So taxonomic composition of Protist. Of course, no, we only look at Protist. We know there's lots of sphagnum reads, but we are not interested in that anymore. There's still Streptophyta in it, but it's not Embryophyta. It's more the unicellular algae. And then we can summarize number of ASVs, number of reads, and then sort them in the decreasing order. So we see we have mainly ciliates, then ochrophytes, then chlorophytes, reads, and so on and so forth. And there's 20 more rows. So when you plot the taxonomy, you don't want to have necessarily all groups plotted, but only the most abundant ones, and the other you will store them in other groups. So for that, you need to select them and then create summary table with this most abundant taxa. So again, I group my taxoprotist table based on phylum kingdom. I do the same summary as before, and then I change the taxa if the rank. So I will only put on the graph the 10 most abundant clades of phyla. So if you are at row number 11 or more, then just change the taxa name to other Protist, and otherwise use the phylum. So here we don't see because all the phylums are from the most abundant one. But here you see all the low abundant phyla. They will be renamed other Protists. So that's Protist taxa. It's only at the taxa level, so it's only 30 line long, so one row per phyla. If we want to have that for each ASVs, we have to create a new table with assigning this taxonomy to all of the different ASVs. And of course, so we still have the issue of compositionality. So we can do some transformation to take into account this bias. Most of the time, we will use square root transformation of the relative abundance to compare the relative abundance between samples. So it's named Hellinger in the decostant function. So it's square root of the relative abundance. You see that the sum of relative abundance is not equal to 1 in all samples. But as you square root way after, then you change the sum per sample. And you see that you see that kind of correlation between the number of ASVs and the transformation of the sum of the transformation of the read count. So it doesn't totally remove also the sequencing depth bias in that case. And that's why we haven't used that for the alpha diversity. But to have an overview on the relative abundance, it's already enough. There's actually more way to transform your data and take into account this compositional bias. So there's multiple other tools. So particularly if you're interested in change of relative abundance of certain groups, and you want to statistically test change in relative abundance, for example, of ciliates with two types of habitats, then you need to use special tools to take into abundance and to account the compositional bias, which I won't show you. I can add at the end of the script in more resources. So using this transform data, we will sum the relative abundance per sample per taxa. So again, I take the matrix, which will transform into a data frame. So here we still have the Protist OTU matrix with read counts in it, just to show you the 10 first rows and 10 first lines of this element. So we still have the matrix, but the first column is the sample names, which we pivot into a longer data frame, just as we have done before. So to have this relative abundance square root transformation of the relative abundance of each ASVs in each sample. We again filter zero counts, and then we add the taxonomy of the different groups. And then we can summarize per sample, per taxa, how many ASVs of ciliates we have in sample 1, how many read counts we have of ciliates in sample 1. Or here for Apicomplexa, so in sample CB1, we have 4 ASVs, and the sum of square root transform relative abundance is 0.11. You can also summarize your data per habitat. So once I have average per sample or sum per sample, I can go back and know which ASVs are shared among the samples from the same habitat. So that's why I have to redo the full process from the matrix. Just to show you the table. So now I've just summarized the reads per habitat and per taxa. And if we plot that, that means doing a bar plot, just as we have done before with all the main groups, we will now have the Protist taxonomical composition per habitat. And you see that there's no huge variation in relative abundance when you sum over all the samples, except for the pool, which seems to be a bit different. So more Opalosa reads, for example, perhaps a bit less chlorophyta. Again, you can save that into a PDF if you want to put that in your nice paper. So if we want to have both reads and OTUs, we have to first average the proportion of ASVs over each habitat. So again, if we start from the matrix, we pivot longer, we add the taxonomy of each ASVs, we add the habitat type of each sample. We here group the value by taxa, habitat, and ASVs. We compute average reads of each ASVs in an habitat. And then when we group again only by taxa and habitat, we can count the number of ASVs in an habitat. So the total number of relative abundance and average count in an habitat. If we plot again, doing the same, so now we can split between ASVs and read count, which are not dramatically different, which might also tell you that you have still this bias of more reads, more ASVs. They look like really well correlated. Save to PDF. Yeah, of course, if you want to do differential abundance analysis, it's great to have dedicated analysis. I will find you the reference at the end of this session to do this correction. Great, let's go for beta diversity analysis. So most of the time we use the Brakertis dissimilarity index. That's relative abundance-based dissimilarity. Of course, you can go for occurrence-based dissimilarity, but as we have pseudo counts or pseudo quantity, we can actually use them, and it's worse to use the relative abundance after this square root transformation. You get better signals like that. So the easiest analysis is to do an NMDS to get the first overview on how similar are your data. And actually, in our case, for the Protist, we don't have any convergence after 20 try. So you can try to let the NMDS computation have more tries, so to go to 50, but still we don't have any convergence. The stress is much too high, above 0.15. So one way to go now is to add a third axis. And you see when you add a third axis, then you reach more easily a solution. Of course, it's more complicated to display because you have no 3 axes and you need to do 3 plots with axis 1, axis 2, axis 1, axis 3, axis 2, axis 3. So it's a bit more tricky, but there's no other way around. So if you call an NMDS object, it will tell you it's based on 3 dimensions. The stress is also not really optimal. Optimal stress will be below 0.1. But still, we are below 0.15. It's okay. Above 0.2, it's really questionable, and above 0.3, it's totally random. So always look at your stress and also write down the stress value in your NMDS to give more support to your analysis. And actually, Vegan have an internal plotting function for NMDS, but I will show you how to use actually ggplot to plot that. So in the point object of the NMDS object, you have the x, y, and z coordinates of these points. So you can actually put that in a data frame and then use it with a ggplot to do nice graphic, which I'm doing here. I do a nice data frame, and then I do my plot. So here on the 2 first axes only, MDS1, MDS2, I'm plotting with points. The points shape would be dependent on the peatland and the color dependent on the habitat. And then you can add the ellipse, which summarize how much space they use in these multi-dimensional scalings for each habitat. So how well are samples from the same habitat clustered together. And then you can add some annotation, like for example, here the stress, which you can extract from NMDS stress. And the annotate function is handy to annotate any plot with any text. So here is more nice NMDS. So the points are not really big. So if you like to increase the size of the point, we can already see that there's some sort of grouping per habitat. So the purple pool samples are more here on the down right side of the graph, while the hummock, which is more dry, are more on the top left part of the NMDS. So again, for those who are not really used to see such graphic, more a point is far from each other. That means more the 2 samples are dissimilar. Of course, here we don't see all the variance because we have 3 axes. So it will be necessary to plot also axis 1 against axis 3 and axis 2 against axis 3 to grasp all the variance of the multidimensional scaling. So now we want to test whether we have good environmental data. So we have to know whether environmental parameters, which are actually the factorial, we don't need to treat that. It's the amount of nitrogen, carbon, pH, and the water table depth. So for those who are not working with peatlands, it's a common measure in peatland to measure how deep is the water table, so the water level from the top of your sampling position, so from the surface. And it's a really a good parameter to estimate how dry or how wet is your habitat. So to know if we have good environmental parameters, so actually the numerical ones, the quantitative, you can do a Shapiro test. So the Shapiro-Wilk test, which is testing the normality of the distribution of the values. And if you have a significant test, so it will reject the normality of the distribution of the data. That means in that case, here for the nitrogen, it would be great. It's a good practice to transform the data so that they find the more normal distribution. And one way to do transformation is log transformation. So we can do log transformation of the values. Here we have put in this test. And we see that great for nitrogen, log transformation works well. We cannot reject the normality of the value, so we are fine. But for the other parameters like C, pH, and water table depth, we are still have significant p-values, so we don't have good normal distributions. So it can take a while to search for all possible transformations. So typically you can square root, you can power of 2, you can do lots of power transformation actually. And for that, you can use scripts so to test which transformation is the best, and use the W value of the Shapiro-Wilk normality test to rank the transformation between each other. You can also use the AKK information criteria to compare the different transformation and the best fit later on. So actually, I've wrote a function to find the optimal transformation, which is inspired by another one. So if you go in the files you have downloaded, you will find this transform 2k2r, and it will actually test with exponent -2 to exponent 2 by a step of 0.05 all possible transformations. So x to the power of -2, x to the power of -1.95, and so on and so forth. Compare all these transformations with this Shapiro-Wilk test and keep only the best one. In the original package Air Companion, they are also doing that, but I think they don't give you which transformation they use at the end. So you are losing this information. They just give you the transform data as output. And that's why I rewrite the function myself.
Guillaume, I got a question here about the homoscedasticity and also the residues testing. Do you test that for PCA or NMDS or not?
You can test that most of the time. Doing the normality correction is easy enough. Once you have corrected your value, you can test that, and normally it should improve the homoscedasticity. More your data is normal, more the distribution of the residual is better. I don't go that deep, but you can definitely test that. Sure. So now if I apply the function to all of the values, so you will see for all parameters, you will have the transform values, the type of transformation used. So here it's for -0.05, the W score, and the p-value, so which reject the non-normality of the data. For C, we have for -2, for pH also. So here we still have a p-value which is significant, so that means transformation is not really the best, but it's the best W we can get with this data. Here we can reject the normality, and here also with another kind of transformation. So
I got another question. Is there a function giving QQ plot for each of the lambda from 2k ladder of power?
No, because I actually have write this function myself. So if you want to add the QQ plots and look at the correct distribution of this data, then you have to implement that yourself. Sorry. But maybe that some package have already implemented. So, but I don't know that. So now I just apply the transformation to the environmental table in db-RDA style, so not anymore with the apply, but I pivot my environmental table to a longer data frame, group by parameter, apply the transformation into a new column named trans, and then I re-pivot the table to the wider format. And now we have the same environmental table, just that here the value for NC, pH, and water table days, they are the transform values. And you can plot the distribution of your values, if you like, across habitats. So here it seems that C is not significantly different among habitat, while N, here water table depth may show some. So of course it's expected that in the pool the water table depth is much lower than in all the other environments. So that is normal and expected. Here the pH also might change from forest soil to the pool. So nothing really unexpected. And if you want to test for the significant difference, you can apply exactly the same test as for alpha diversity. So fitting an analysis of variance and doing the post-hoc TK test. Well, and then you can use then the transform environmental parameters to fit to your NMDS. So you can create a function with unfit. So it's a function in vegan, which will fit your NMDS with different parameters you have in your data table. So when you do unfit, yeah, it's crucial you have the sample in the same order. So the sample in the matrix you have used to create the NMDS need to be exactly in the same order than in untrans table, because unfit and all the vegan function, they never checked for the sample names and the sample order. So it's your task to be sure that samples are ordered the same in these two tables. And here in our script, actually, we have taken care of that earlier when we filter the data. Actually, we have filtered when we create the matrix. We have used the sample names as they are in the environmental table to subset the matrix. And it will take the exact order of the sample in this env-AB to subset the matrix. So doing that, we are sure we have the same order in both the matrix and the environmental table. Well, let's go for the fit. So we can have a look to the output. So it fits the vector, so the numerical parameters. So you see that water table depth have the highest explanation power, is significantly explaining change in Protist community composition, and N have marginal significant effect. And the two other parameters doesn't seem to be significant. Also the peatland, it's good news. We don't have effect of peatlands. It should be considered as random effect in this analysis. And on the contrary, the habitat seems to have a huge effect, just as water table depth actually. I'm not peatland ecologist, but most likely the habitats are also defined by the water table depth. So it might be exactly the same using habitat or water table depth. And now we have the NMDS fit object that we can save into a data frame. So in the object, we have the arrows. If you want to draw arrows on your NMDS, so it's the X and Y position of these arrows, which are actually, if you want to plot them, then you have to multiply them by the square root transformation of the R square of each parameters. That's also the standard when you plot with a vegan package. Let's go for the plotting. So we just reuse the plot NMDS used before, and we add just the fit we just wrote down. So here you see that the text is not placed really correctly. So either you place them one by one and you make a nicer figure, or you use a package which is doing that automatically for you. Its name is ggRepel. I will place annotation actually on position and go back to the plotting. So here, in place of using geom text to place the text at the end of the arrows, I use geom text repel, which is actually the same function, but with repelling automatic placement of the label just to avoid overlapping and to be sure that the text is placed inside of the plotting region. So it's actually all the rest is the same. And again, if you want to save this figure into a PDF, just use ggsave. So at the end of this first analysis, we see that water table lakes explain the most change in the community composition, or seems to explain the most change. We can do more plotting and more testing. So unfit again was only done against axis 1 and 2. With the option choice, you can change the axis you test, and you will see whether you have better fits of the other parameters along the other pairs of axes, so 1, 3 or 2, 3. Great, but actually to do more formal statistical tests, you need to go for PermaNova, so permutational analysis, multiple analysis of variance. So the function is Adonis to do this PermaNova, so you can go in the help to learn a bit more about this test. So here we want to test a change in community composition due to habitat. And here what it tells you is that almost 20% of the variance is explained by the habitat with a strong significant difference. So the R square is different to NMDS because NMDS is like multidimensional reduction of the variability you have in a Biais dissimilarity matrix. So it doesn't work exactly with the same values. And actually, when you base your test on Biais dissimilarity, you also lose a bit of variance because you have only one dissimilarity value between two samples. If you want to place each sample in a more complex multivariate analysis, you have to do a CCA analysis. Just before going for CCA, one thing to consider is when you have more complicated experimental design. So here we have habitats, but they are found in multiple peatlands. And we don't want peatland to interfere in our statistical test. So in Adonis function, you can use the strata option, which will constrain the permutation inside each level of this constraint. So here we'll do a test for each peatland, actually, whether habitats have an effect. In the end, it doesn't change anything, but if you have block design or nested design analysis, it's really important to use that. So you can either use strata, and if you have more complicated design, you can explain how to do the permutation with the our function. You can go in the help of our function, and here we use actually the blocks, so which is actually the same as strata, and you will see the result is exactly the same. But if you have more complex, nested, and interrelated samples in your design, then you have to go with the our function to constrain the permutations. So last step is doing the CCA. And with the CCA, the advantage is that we can remove the random effect of the peatland by partialing out the variance, which is due to each specific peatland. So we build a model which is based on the Hellinger transform matrix. So we use the full matrix here and not only the Biais dissimilarity. Otherwise, it's a dissimilarity-based correspondence analysis, which is not exactly the same. And just like in unfit before, we have a formula which will search for explaining parameters. So with nitrogen, carbon, water table lakes, and pH. And by adding these parameters, so condition, it will remove the variance due to, if you have transects, for example, that's what you want to use to remove variance due to your different transects, which are random effect parameters. So let's run the model. It's really fairly quick, and then you can do an analysis of variance of these models. So as in the analysis of variance, you do permutations. It's maybe a good practice to set the seed that the departing of the permutation of the random algorithms so that you can always have the same result or same R square every time you rerun the same script. And you can summarize the analysis of variance into a data frame. So again, here we have p-value. really significant for water table depth, also significant for the nitrogen, but not significant or marginally significant for pH and not at all for C content. So now we have built a model with all the parameters, but we can try to optimize our model and selecting the best model. And there's two ways we can do for that is to build stepwise forward or stepwise backward selection. So for that, we need a model, initial model, where we don't have any parameter to constrain the CCA. And then we use the function ORDISTEP, which starts with the model without parameter fitting or constraint, and then we'll use all the parameters in the first model to find the best model with the lowest IKK information criteria. So we will print all the steps he computes, and as soon as he don't find a lower IEC, then he will stop. So it can take a while. So here we don't have a really big data set, so it takes not so long. And at the end, you see it ends up with the last model was with water table depth in nitrogen content. That's the two parameters, and you won't use C and pH, more or less what we expected. You can print again, and here you see the formula. You can see the adjusted R square of your model. So how much variance, total variance is explained by the model, which, if I remember, is not so high. So only 2% of the total variance, which is really small. And you can also try to go backward with backward selection. So the way to write that function is a bit more complicated, but actually it starts with all parameters and remove one after the other and test the AKK criteria. And here in the backward selection is ending up with water table depth only as explaining parameter.
Yeah, maybe Guillaume, I can just add something important. Here we have just a few variables, but what is really important before doing all these different steps is to check if there is some correlation between your variables and remove the variables that are correlates before doing all these steps. So this is a step that Guillaume didn't explain because it was not our case with this data set. But I know that for some data sets, you will have like 20 or 30 different variables, but most of them have correlation. So it's really important to do this step before doing an ORDI step or doing ordination.
But it's important that you mentioned that, because to optimize a model, you need to find the lowest number of parameters which explain best your data. So here the model 3 have even lower adjusted R square, so we can actually skip that part and just do the ANOVA and the fitting with model 2. So the analysis of variance is actually looking at all terms of the formula to analyze how much variance is explained by these parameters. Here you see that they only partially explain the total variance, which is here the sum of all these three values in the chi-squares, but they still have really significant explanation. So this model, so CCA, actually you can extract the X and Y because you have two constraint parameters. You have two constraint axes, CCA 1 and 2, and then you have multiple other unconstrained axes, so 57 here, that you can use also to plot, but here we will plot against the two constraint axes. There's also, as it's all vegan functions, you have an internal vegan plotting function. So it's the standard plotting of vegan, which is not so nice. That's why I was moved to ggplot. So using the scores in the biplot, you can also extract the position of the arrows for N and water table depth, and then use that for creating your figure. And now you see a much better separation of the habitats along the x-axis, because that's the axis fit, or we constrain with this habitat parameter. And on the y-axis, you have the constraint with the nitrogen content. So now it's a bit easier to differentiate your samples from habitat, and you see that really pool habitat is totally different from the 4 others, which are more terrestrial, while the pool is much more aquatic. So it's describe your habitat quite nicely and correctly, as expected. And then you can see that there's probably a nitrogen gradient from transition to forest to lawn and to hammock, which seems to drive also the Protist community composition.
Yes, maybe can you just show how you can compute the percentage of variance of each axis?
It's actually the person of chi-square. Yeah. So if you sum all these 3 values and you divide one value by the sum of the 3, then you get the percentage. Or I guess it's that. Or you can actually, you can also take here the eigenvalues of the axis. So the total inertia is 5.6, and the first axis, the eigenvalue is 0.16. If you do a percentage, so no, the first axis explains 2.9% of the variance, and the second axis will be around 2. Yeah. So if you like, you can use these values to compute the percent variance explained by the axis. But you have lots of unexplained variance. That's not much of a surprise working with Protist, because you have so many different functional groups that they will react differently to different environmental parameters, and it's hard to find a good model explaining the distribution of all Protists at once. Great, and I'm done just in time.
So yeah, well done. That was a really cool presentation, and I think now you understand that this kind of data set are so huge. It's sometimes difficult to find the best way to present your data set. So just before the question, I can maybe just show you 2 things on my screen. I will share my screen because yes, this study, we have 1 publication using this data set, and we didn't study the whole diversity as Guillaume presented, but we focused only on 1 group, and we focus on the OOMI sets. It's sometimes easy just to study in details 1 group, then wanted to study everything and be lost in the middle of your data. So we use this data set and we extract only the OTUs related to OOMI set, and we go into details and check carefully if it was really an OOMI set, and we check the distribution in the different microhabitats. And that was my second publication. And here we found also that in pool, there is a different community of OOMI sets, with some OOMI sets that are really specialized in pool. And that makes sense because it's a freshwater organism that you can find here. So that's the first publication we made with this data set. And we also work with Christophe Sepe, and he has a chapter of his PhD that are related to this data set, is chapter 2. Yeah, so he did some kind of preliminary test on using NMDS and some NDCs, and we tried to compute some co-occurrence networking, but it was really some preliminary results using this data set. So it's why we didn't publish yet the whole data set. So it was a great opportunity, this workshop, to just share with you this idea and how we will maybe potentially publish these things in the futures, even if it's a bit tricky and the data set become old. As we have explained, we sequenced that like 7 or 8 years ago now. So it's quite, yeah, everything is moving forward very quickly, but still there is some valuable information in that data set. And I hope you learn some stuff with Guillaume on how to treat these things.
Yeah, question of the Adonis function. You explained, Guillaume, that you can use the blocking thing with the strata and permutation. Can you also use just a plus, like you would do with the LM function, to have block plus then the variable that you use, and then you do a sequential two-way ANOVA. Is that similar or not?
Actually, if you add the parameter in the function directly, so that means you write the function in function of one parameter and your blocking parameter. That's what it will actually test the effect of this parameter too. So it will tell you if you have an effect of your transect, for example, or the peak length. But it will not constrain the permutation when you do the statistical tests, and it's not equivalent. So when you are testing for your transect effect, you are not constraining the permutation of the sample inside one transect. I think it covers different cases. It's just you test whether your design have an impact on your community composition or not, which you should actually not be interested much, or you suppose, okay, your experiment is designed well enough to not impact much on the experiment, on the data, on the results, but you can still control for the random effect produced by multiple replicated experiments. If you have replicated 3 times or 5 times the same experiment, you still need to take into consideration that block design. Yeah.
I have one question about numbers or ASV numbers. If I understand correctly, you told that if for each group rarefaction curve was not saturated, you cannot compare numbers, yeah, for ASV numbers. But if it's the case, it's no problem to show these numbers in publication that show that taxa or ASV numbers were high in one habitat but not another, but you need to show then saturation curves for each group, yes?
Okay. But as I say, sometimes it's misleading to show rarefaction curve because you have removed the low abundant ones, the low abundant ASVs, and your rarefaction curve will always saturate. As soon as you remove singletons, doubletons per sample, then all the rarefaction curves will saturate. But it's it's just because you remove this low abundant.
And another question about why you decide to show today square root transformation. Why this one? Just I'm curious because there are a lot of different and why was this one which you highlighted?
Yeah, because it's commonly used. Actually, it has been used a lot for correcting the relative abundance. I haven't tested or show here a test that it's correctly correcting the relative abundance bias, and it's also correcting the exponential amplification bias of the PCRs. But whether square root is better than log, if you don't have data, you know the exact relative abundance. So like mock communities, you cannot say which one is the best, and most likely it would change from one data set to a data set, whether you have 35 cycles in your PCR or you pull your sample to create libraries, how much read you have per sequence. All that will potentially influence how much relative abundance you get from one ASV in one sample compared to the original abundance of these organisms. And last but not least is also that some organisms can have multiple copies. So like Enrique showed this morning, for SSU, it seems to be correlated to the size of organisms. So we are lucky with that. We can probably correct relative abundance based on the organism size, but it's still work in progress. So.
But did you try to compare central log transformation to square root transformation?
We can do central log transformation. There's multiple other transformation. Yes, I wanted to give you some reference to this. If we don't have mock community, we can compare. So we cannot say this one is nearer from the reality than the other. And it looks like in the last years, people tend to say center log ratio, it's better for compositional data, but what it's true for meta transcript, so gene expression, transcriptome datasets. And now we realize, well, metabarcoding data is also compositional data. So let's take the same. And the last paper I've read on that topic from this year, from last year, they say actually we might not necessarily need the center log ratio. It's not necessarily the best transformation. Actually, some people say rarefaction, it's still okay. And yeah, it's still an evolving topic because the method actually was developed for transcriptome analysis and gene expression analysis. It not necessarily apply one-to-one for all type of data, but we surely have an issue on the abundance data, and we need to think about how to.
And as you said, the rarefaction, you will lose a lot of reads depending on your data sets. So by losing all this information, sometimes it's just you throw away 90% of your data set, and it's not that good. So it's why doing this, using this residual of linear models could be a best option. But that's true that if you want to interpret the value of your alpha diversity based on this residual, it's a bit more complex to use, as it's not a number of ASV anymore. But it's another way to provide information about your diversity.
I have a question. It's related to correlations. As David said, when you have many, like 20, 30 parameters that could potentially correlate, you can, of course, check what is the correlation between them and set a threshold. And first question is like, you can use what threshold, what you consider as being a correlation that is too bad to be used. Is there a way to that would be specific to your model when you can say, okay, the threshold would be at, I don't know, 0.99, 0.95, 0.9. And then how to deal with this, like just to choose one of the parameter to put them together, or like to use the axis. And if it's to use the axis, if you have any references on like maybe more like the script to select the axis of a model to say, okay, this is my axis for these 4 variables and this is the axis for the 2 variables.
So yeah, there's when you have many parameters, yes, you have this different way of going. Either you do correlation, so that means Pearson-Spearman correlation. You look at what is significantly correlated together, and you take only one parameter out of the one which are correlating together. Another way, not using correlation, but you can use directly the CCA. You can create a model with all the parameters, and then there's a tool in vegan which is called VIF CCA. It will look at the in the CCA how much the parameters are actually correlating in the same direction. It will check each pairs of parameter, I think, and it gives a value per parameter. So, and if a parameter have a VIF value above 10, I think that's in the guideline from vegan. They say if a parameter have a value of 10 or more, it doesn't bring more explanation in the variance of your CCA, so you can remove it safely.
Okay, but this you do before the forward selection?
No, you do a first CCA with all parameters, without ANOVA fit or anything else, and yeah, like that, you keep only the parameters which have a value below 10, and yeah, the function is doing the selection for you. But to come back to the comment of Estelle, yeah, the last way to go when you have correlation between parameters is not necessarily to pick one, but to group parameters which are actually explaining the same thing, like which could be correlated to the moisture of your soil, for example. And more you have moisture, more you have available carbon, for example. you could have multiple parameters directly connected to the moisture content, and then you can do a PCA with these 2, 3, 4 parameters which are actually correlating with each other, and pick the first axis of the PCA, which will summarize the variance of these multiple parameters in one composite variable. And you use this composite variable then in the CCA model. I give you the reference. So in this text file, I just add on the drive some more references. The first reference where they do also they use the residuals of this linear model of alpha diversity against sequencing depth. They also group these parameters and do a PCA on them, and just use the first axis of the PCA as a composite parameter for beta diversity analysis later.
If I can add something about this, because sometimes the first axis is not doesn't represent the highest variance, and I used to use the beta disp function to take the distance to the centroid. So I also have a synthetic information on the 2 axes.
Okay.
I don't know, it's something I did already.
All right.
Because I was not happy with my first axis.
Yeah, if it's only 10% of the variance, then
or less.
you
But
information.
Sorry, could you just repeat what you are using, Florine?
Yeah, it's beta disp function, and it's from vegan, I think, the vegan package, beta disp. And it measures the dispersion of your samples to the centroid. So it measures the centroid of your clouds and then give you this distance, if I remember well. Yeah.
It's beta disper.
Beta disper, thank you. Yeah.
Thank you.
Yeah, it's to measure the dispersion.
Okay, and you talk about CCA, but it's working with CCA, RDA.
Yeah, so basically RDA and CCA could be more or less equivalent. You will more use redundancy analysis, RDA, for when you have a lowest number of parameters. So if you have a small design, like 2 parameters in 2 different habitats, you will more go for RDA. For CCA, it's more when you have multiple sites, multiple parameters, multiple it's a more complex design.
But about what you said, because RDA is based on Euclidean distance, and then I thought this is not appropriate to handle this species community matrix, because I've always used this db-RDA when you can choose your distance metric, so Curtis, and then do the unconstrained or constrained ordination, but not with Euclidean distance, as it is by default with RDA.
Yeah, so I had also different view. I think once I had comment from Legendre was kind of he developed these methods, and he still say that CCA and RDA it's more powerful than db-RDA because you use all the data at the end, so you use only the dissimilarity matrix. When you use a similarity or dissimilarity index, you have just compress all the differences in one value. CCA and RDA, they are taking advantage of the full matrix. And even if they are based on Euclidean distance, they are using it also between all the ASVs or OTUs in the matrix. So for me, CCA and RDA still are more powerful than db-RDA. So when you are working with phylogenetic dissimilarity, for example, you don't have much choice than using db-RDA, because the only dissimilarity value you get it from a unifrac, for example. But when you have the OTU matrix, then it's better to use it directly in the CCA.
I have a question regarding the beta diversity when you do the non-metrical multidimensional scaling. You said that to make your stress level, you increase the number of axes. Like if you still have a really, really high stress level, you have to keep on increasing the number of axes, or you can increase the number of tries?
You can increase number of tries, but most of the time it's not enough. So that was also the case today. I've increased tries to 50 and you still not find any convergence in the solutions, and still with high stress values. So above 50 tries, there's no need to go above 50 tries. If it doesn't find convergence after 50 tries, then with 100, with 500, it will still stay the same. So really the best way is to go with more axes. And if you have more than 3 axes, that means your data is really complex.
Okay.
Perhaps you have too many parameters, too many sample types.
That was my next question, how to interpret, like when you have more than 3 axes, because you have to still combine 1 with 2, 1 with 3, 1 with 4, and then like all the combination to make the interpretation of those results is sometimes really complex.
Well, so one thing which happen often is that you have one parameter which explain most variance, or sometimes it's one of your design parameters, so the habitat type or the experimental one parameter in your experimental design. So drought, or when you simulate drought, or when you water, and once you have found these main explaining parameters, just use per MANOVA, you show, okay, 60% of the variance explained by this parameter. Well, if it's a factorial parameter, then what you can do is to do separate analysis, and inside the different factorial parameters, just split your data, and then you reduce the complexity like that, and then you will concentrate on more specific environmental parameter which can explain the change in your communities. The other way is rather splitting samples, it's splitting by taxonomic or functional groups, so large functional groups, which is actually what we have done today. We are selecting only the Protist out of the dataset. You can consider Protist is still too huge. It still cover almost all functional groups you have in the split lens. So let's go and split them further to the different taxa. By doing that, often the problem is that some groups you will not be able to use them because they are too low abundant in the dataset. They are not well covered by the V4 or the V9 primers, so you will only limit the analysis to certain groups. The one which are well covered, like Cercozoa, ciliates, amibs, and they might have different behaviors, response to the habitat change. Some might be same along the water table depth gradient, some might change strongly, like we know the tested amoeba, they are good biomarker of this water table depth.
Please just change, like if you don't get like enough results, or if the stress level is too high, you just keep on reducing like the complexity of the data.
It's also depending the story you are, or the hypothesis you are after. So if you are interested in special functional groups, or if you just want to know, well, with a drier habitat, we have more diversity, or with a more rich habitat, we have higher diversity, or the composition is much more different in drier habitat than in wetter habitat. If we have more redundancies in the community composition or not, how the community can react to stress, things like that. So it's really depend the question you ask with the data also. One general comment is when you publish your script, or when you attach the analysis script to a publication, it's a good practice also to add these session infos with your script, so that you have all the version of your R and your packages.
That's something I request every time when I review a paper, is adding the version of each package and R. Because if you want to redo the script, even if you don't have the script, if you have the package, you still can redo the things basically. But yeah, it's always great if you can provide the whole script. But I would say, I must admit, I don't do that every time. It depends on the paper.
For Cutadapt, they have it evolves a lot in the last years, and and if you don't use the same version, probably you will not demultiplex the same or not remove the same amount of primers.
It's really a powerful tool, but it takes a bit of time to understand how you should put your things together. But once you manage to understand how it works, you can do basically anything with this Cutadapt. Yeah. And it's very fast also.
Yeah, that's crazy. You don't need a big server to run that.
It's about what you proceed in your script. When you compare the species richness versus the number of read, there is a strong relationship. This means that there is a bias, and you use the residual to correct this bias. Could you please explain me in detail your script and the formula you use to correct this bias, please?
Yeah, actually, I'm just plotting the read counts against the alpha diversity values. In the first plot is the richness in the y-axis, and you can build actually your linear model with the total read counts or the log correction of these read counts or the square root correction transformation of this total read counts. So if you want to have an easier or simple way to manipulate that, so only for richness. So richness, you can get it with spec number. So not Protist. Spec number gives you the number of ASVs in each sample. If you do the row sum of your matrix, then you get the number of reads. That's the two set of values you are working with. And then you can just put that in a data frame. So we have a nice data frame, richness reads, 1 column for each. And we can even push the row names to the column name sample. So that's a habit I have with when using db-RDA, because when you pipe or when you use further function, then you may lose the row names. So it's a good habit to have to move the row names into a column. Great. So we have that values. We can save that into a rich table. And then you can create a linear model. So you want to explain the richness by the number of reads using the data rich. So that's the linear model. You can save that into temporary linear model or rich LM if you like. And if you use the summary function, it will run an analysis of variance on the linear model, gives you the fit of the two parameters. So the output of these summary functions will give you some R square, adjusted R square. So you see that the total number of reads explain 56% of the variance of the richness. You get the estimates of the slopes and the intercepts of your linear model, and also the p-value of the slope and the p-value of the intercept, which is, recall here, the p-value of the slope. So it's the p-value of the linear model. Well, to get the residuals of your linear model, you just have to use functional residuals, which give you something like that. again, you can save that into your data frame. And then if you want to rescale your residuals, so now it's not really the best practice that you have res because you don't have the sample names attached to it. So best will be to add to the rich data frame. So you can also do that directly in db-RDA. So we reach with the residual, then you have to create a new column of rich that you call res. So here you don't need to say data because you're inside the db-RDA function, and you use richness and reads as the column names of your input data, which is here. And if you want to rescale the res on -1, 1 scale, I think the formula is 2 times and then you can summarize your values. So the residuals variance vary between -73 and 73, and the the residual variance of the scale, the residuals vary between -1 and 1. It's just easier to compare like that between the different alpha diversity indices. But as told before, if you use the Hill numbers, you are also already on the 0, 1 scale for all Hill numbers, and then you can do also the linear model with Hill numbers. But that's next level.
You mentioned there are many methods to deal with compositionality of high throughput sequencing data. And we can't say which one is better than others, right? Or you have some suggestions currently which method is better than others deal with computation. Yeah.
It will depend which statistical test you run then. So there's no one correction you can apply at the start of your script and then forget about compositionality. That doesn't exist. So you have to adapt the bias correction to each type of analysis you are conducting. In the Glor et al. Paper in 2017, so the reference is in this text file I just add in the drive in the folder statistical analysis. They are giving some advices of what to do for different kind of analysis: alpha, beta, network analysis, co-occurrence, differential abundance analysis. It was almost 5 years ago now. In the meantime, it changed a bit, the recommendation. So my recommendation, if you want to do differential abundance analysis, is to use this ANCOM-BC tool, ANCOM-BC tool. I've put the reference also in this paper. That's the third reference, Lean and Pindada 2020. And this one is dedicated to differential abundance analysis and not for all the others, not for co-occurrence, not for alpha or beta diversity analysis.
Yeah, I have seen the ANCOM-BC paper, and I saw the authors also make ordinations and to see whether the ANCOM-BC normalization method can well separated the community, different community. Yeah, they use the NMDS plot.
Okay.
But if I want to compare the different normalization method, like I compare the Glor's CLR transformation method with the ANCOM-BC method, I should use the mock community, right? I can't use my own data.
Yeah, of course, you need to use a data set for which you know the ground truth, or for which you have another way to measure the relative abundance or the change in community composition.
So I should create the mock data by myself, or there are some standard way or resources we can download some known data?
There is multiple mock communities, of course, and they are actually using always mock communities in such papers where they present a new tool to test whether they perform better or less better than other tools. So you have already tons of mock communities, but mock communities are often only good to test this Bayes in relative abundance and not change in community composition. You need to have multiple mock communities with, I don't know, hypothetical design that I don't know, you have more Cercozoa or more of certain taxa in one sample compared to the other, to have complete set of simulated data which are related to natural factor or to unvolatile parameter is quite hard to find. So I think this mock community works well, yeah, for the change in relative abundance, but not necessarily for beta diversity or more complex beta diversity analysis. So it will be still hard to know which tool performed best, and at least it performed better than not doing any corrections.
During the filtration steps, we tried to get rid of ASVs which occurred less than 4 times. This was one thing. And the other thing was to remove samples with low read count. Is it correct? I didn't understand, are we really removing whole samples? And if so, what was the criterion? I'm sorry, I didn't understand that.
So actually this sample was really, it was due to the low amount of reads in that sample. So it have 25,000 reads in total, and all other samples have more than 100,000 reads. Actually, I haven't shown that, but when you remove all the sphagnum or the macroorganisms sequences, so this sample also had a really low amount of reads for protists. And the risk is that you compare samples not covered at the same level.
Yeah.
Better than keeping them in the analysis if you have enough replicates, or if it doesn't impact your statistical test, then it's safer to remove them.
Okay.
And in particular, if you're doing rarefaction, in that case, it's really critical. So rarefying to 100,000 or rarefying to 25,000 have, a big impact.
So the criterion is to have a similar number of reads, but there is no logical or strict criteria.
Actually, you can do an outlier detection analysis if you want to have a statistical test which tells you, okay, this sample is really different from the others. These usually, I could say, okay, all samples are not that much different from the others, but this one was really an outlier. It had 4 times less reads than the next sample with the lowest amount of reads.
Okay.
You can plot the sorted number of reads per sample, and you will have kind of all the same, but just a few samples that are very, very low. And this is the one you will see directly, the one you should remove, because they are too far away. So that means
remove or resequence.
Or resequence, but that means there is a few different explanations why you don't have enough sequence. And basically, the best explanation is you don't have enough PCR product to reach the same level as the other sample. But you try to put all your PCR product to sequence, and that was not enough, so you don't get the good number of reads. So that's the best explanation. Or maybe the quality of your PCR product are just too bad and the sequencing doesn't work well. If you have hundreds of samples, there is always 1 or 2 samples that doesn't work properly. So it's fine to remove them. So it's just about the number of reads for your first question. And for your other question, the threshold about the 4 reads per ASV, if it was to avoid the tag jumping, if I'm right, it's just to put at 0 if you have 1, 2, or 3 reads to have an absence of this ASV in your sample. So it's an approach just to try to reduce the tag jumping effect.
Actually, when you do cut adapt after the second round of demultiplexing in the other direction, you still have a noun or an assign, and it might be that there is some wrong tag combinations in these remaining reads and assigned reads. And from this unknown combination of tags or tag combination you haven't put in your mix or in your library preparation, you can try to estimate the tag jump. So
because usually now we put tag in both sides of your amplicon to define one sample. We don't use a single tag anymore. And by doing that, you can really assess the tag jumping by seeing the error combination, because you will create some error combination, and that's the tag jumping.
Actually, this threshold of 3 or 4, it's far from being the best practice. It's
just up to us to define this kind of threshold. So yeah,
it's a bit arbitrary. I clearly admit that, and as long as we have no more information on the tag jump, it's the minimum we have to do to get rid of. You probably get rid of real biological rare ASVs, typically also because in that case you have many co-amplification of sphagnum, and all the rest of the diversity will be really badly covered because most of the sequencing was taken by the sphagnum sequences. But we know that due to the design and the library preparation, they will have tag jump. So we have to be critical about low abundance ASV occurrences in one sample. And to be on the safe side, the best is to remove them. So of course, more an ASV is abundant over the whole study, more it has chance to produce tag jump. Less an ASV is abundant over the whole study, and less it has chance to produce tag jumps. That's something I've shown yesterday in the other room table, and that you can control for that when you have, for example, mock communities or positive controls you add in the sequencing, and you can estimate what is the best threshold using a control. Because if you, for example, add, you're interested in terrestrial protists, and your positive control is done with marine cultures of algae, and then if you find an occurrence of this marine algae in any other of your soil samples, you know it can only be due to the tag jump. And like that, you can estimate how often an ASV, which is not supposed to be this sample, end up in a sample, and it's correlated with the total abundance of this ASV. So you can build more complex model to correct that. But if you don't have this information, then just do at least this basic cleaning of removing low abundance